Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about gene expression, or how organisms are able to regulate making a protein or not making a protein. Now, this is a little bit of an advanced topic, but I think along comes with something that's advanced, something that's very interesting, and it's really intriguing because at the very core of this is how the simplest organisms in the whole world, bacteria, are able to determine when it's appropriate to make a protein and when it is not appropriate to make a protein. And it's like, how do they do that? Well, ultimately it was determined that they're able to regulate their transcription. In other words, turn on and off genes to do that. And it's quite remarkable how we know that. And we've applied that knowledge to ourselves and larger organisms, eukaryotic organisms. But today's conversation is going to be primarily about bacteria, and I find that it's going to be really interesting. I hope you enjoy it. It's one of my favorite topics of all of molecular biology. And so here's a picture of E. coli, and they're very, very small. You wouldn't be able to see them unless you were looking at them under the microscope. And so one of the things about it is we are able to grow bacteria. We're able to grow them on these dishes called petri dishes or petri dishes, and they grow on this food medium called agar. And so back a ways, in 1961, two French scientists were working with bacteria, and they were simply growing them. That's what these little dots are. These are colonies of bacteria. Their names were Jacques, over here Jacques Modon, and Francois Jacob. If I'm pronouncing that right, I apologize. So Francis and Jack, or Frank and Jack, and they showed how bacteria could do this. And what's interesting about it is, let me come over here and show you a little bit about what we're talking about. Let me call up a screen here and say, well, one of the premises was when they were growing bacteria on these dishes like this. Here's a petri dish. And let me give it some food right here, some agar. So this is your basic food. You can add things or not add things to it. And here are the bacteria. I'll make the bacteria red. There they are growing. And what they discovered is when they added, let me go here with blue. When they added something to the agar, in other words, you could add like powder, whatever, like protein powder or amino acid powder to this. When they added the amino acid tryptophan, which I'll just symbolize by tri right there, tryptophan. When they added that to the food of the bacteria, what do you think? If I were to ask you this question, if you add tryptophan, which is one of the 20 amino acids, if you add it to the food, do you think the bacteria should make it on their own or not make it if they're getting it for free in the food? And I think logically you would guess they shouldn't make it. Why would they make it if they're getting it free in the food? And so when you add tryptophan, the bacteria stop making it. And when you do not, let me do this, when you don't add tryptophan to the agar, it turns out that the bacteria, in order for them to live, they have to be able to make their own amino acid tryptophan in order to create all the proteins. And so this is rather interesting to me. It's that the bacteria are able to decide whether or not it's appropriate to make something or to not make something. So let's talk about this for a second. So an amino acid, remember, let me just go up here, is your typical amino acid. It looks like this. They all have this basic appearance, but what differs about them is this R group down here. And so what I'm going to say is, when I'm talking about tryptophan, I'm going to just say that it's going to be tryptophan, like that, as symbolized by the R group. And it's like, well, how does the bacteria go about making an amino acid? Well, it takes something that's not an amino acid and turns it into an amino acid. As long as it has something similar, it can do that. It takes precursors and it creates it in a synthetic pathway, and it happens to be linear. So let me see if I can draw that. So it takes something that's not tryptophan and it turns it into something that's a little bit closer to tryptophan, and then a little bit closer still, and then a little bit more, and then a little bit more, and then over here, let's say you have the amino acid tryptophan. Okay, so this right here, this amino acid tryptophan is what the bacteria is trying to make. But again, here's the thing. When you don't give tryptophan to the bacteria, they should make it. And when you give it to them, they should not make it. Now, how are they able to do that? Well, one way in which they're able to regulate the production of an amino acid is that it requires a protein called an enzyme. Let's just call that enzyme 1, enzyme 1, excuse me, enzyme 2, enzyme 3, enzyme 4, and enzyme 5. Requires all of that protein to make one single amino acid. And you're like, oh, that's a big deal. Well, you need that to live, and so it's worth it. So here's the thing. When there's high concentration of tryptophan, when there's high concentration of it, so there's an abundance of tryptophan, one way in which the bacteria could slow the production, if you have enough, you don't need to make more, is that the amino acid itself feeds back in the cytoplasm, diffuses, and it interestingly bonds. Let me go blue here. It bonds to the first enzyme in the pathway. It feeds back, and when it does is when it attaches, again like it's a magnetic bead, what it will do is it'll alter the shape, temporarily alter the shape of the enzyme, the very first enzyme. And so therefore, it inhibits this enzyme from working properly. And so if this enzyme is not working properly, then this thing goes down in concentration, down, 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 and then down. That's one way that you could regulate the production of a product, is that you feed back and you inhibit the enzyme. But the truth is, that's pretty good. But the bacteria is still, say you're feeding the bacteria tryptophan like in the experiment, you're giving them lots and lots of tryptophan. It's still kind of wasteful to produce these proteins necessary for this pathway. A better way to control it, if you will, would be on a gene level. And so up here is DNA inside the bacteria. Now you know that DNA isn't making a protein directly, it's RNA making a protein. But I'm not going to go into that. I'm just going to say this, that one way to do this is, okay, do you see this right here? Let's say that this area right here, this gene is for this protein, and let's say this gene is for this protein, and this gene is for this one, and this one over here, and then this one goes here. Can you see how there's let's go gene A, B, C, D, and E. There's many genes that operate together to produce tryptophan, ultimately. Of course, it's RNA and then the ribosome to make the protein. So these two French scientists were wondering, how is it that the bacteria are able to stop making tryptophan when you're giving it to them? How are they able to stop making it? I'm not sure. It might have something to do with gene regulation. And so they were thinking that maybe, let me just cut to the chase here. Do you recall the way this works, transcription, is that there's an enzyme called RNA polymerase. And RNA polymerase is able to attach to a region on the DNA called the promoter. And then when it attaches, it moves along, and it starts transcribing these genes. They thought maybe when you give a lot of tryptophan to a bacteria, they're able to stop making these because maybe, check this out, downstream, or I'm sorry, upstream from this operator area, there's another gene over here that produces a protein. I'm going to stay yellow. produces a protein called a repressor protein. I'm just going to use RP for repressor. And when it makes that repressor protein, it makes it, but it makes it in a weird shape. Makes it inactive. And that's kind of odd. So it produces a protein that's circulating in the cytoplasm that doesn't do anything. But how do you like this? When there's a lot of tryptophan present, we go blue with tryptophan. When tryptophan is present, it attaches to the repressor protein, making it active. And so now it has an aggressive appearance to it. And so what it does is it comes up like this, and the repressor protein, now that it's active, attaches to a region right in front of the first gene in the series here, in an area called the operator. And it binds to this operator area, and when it binds, it blocks RNA polymerase from transcribing messenger RNA and then making these proteins. So if you're following this, when tryptophan is present, it's able to block the transcription of the genes necessary to produce the proteins that produces itself. That's very profound, if you ask me. I hope you're following that. And so let's take this conversation over here to the notes, and we'll see. This isn't necessarily the easiest topic we've ever talked about, but again, that's fine. I find that it's one of the more interesting ones, so I hope you're enjoying this. And so, again, French scientists are growing bacteria, and here's a little diagram of some of the things that I was trying to draw. The amino acid tryptophan, there's a biosynthetic pathway that leads to its production. And I was mentioning there's two ways to control the production of a product. The product itself could feed back and inhibit an enzyme called feedback inhibition, or tryptophan itself can actually go back and prevent the genes from being transcribed in the first place. In other words, not even make the protein. And that's even a more profound way to control it. And so, again, Francois and Jacques, in 1961, quite a while ago, proposed a model which is sort of a diagram of how they believed bacteria are able to do this. And they thought that there's several genes, like we saw A, B, C, and D, there's several genes that are important in the production of enzymes in a particular pathway, and we're going to call those structural genes. And they thought that all these genes sort of work together. There's an area called the promoter where the RNA polymerase binds. There's the operator sequence where the repressor protein binds or does not bind, depending if it's on or off. They felt that this whole region should be called an operon because it all sort of operates together. And it involves three elements, sort of this control area, structural genes, and what they're not showing in this simple diagram is the termination sequence. So it's promoter, operator, genes, terminator. So let's look at this tryptophan operon. So in other words, these are the genes that produce the messenger RNA, which produces the enzymes necessary to produce the amino acid tryptophan. So way upstream from the promoter of the trip operon is this regulator gene, or TRPR. This makes messenger RNA. This makes an inactive repressor protein. So this thing's cruising along. So if it's inactive, what does this tell us? That most of the time, bacteria make tryptophan. You're like, well, I'm not sure I saw that. Well, RNA polymerase, which is shown here in white, normally attaches to the promoter. It cruises along, makes messenger RNA, produces the enzymes. The bacteria makes tryptophan. So the bacteria is making tryptophan all the time because the repressor protein's inactive. But what happens if you give the bacteria tryptophan? You want to shut it off. Shut it off. We've got free tryptophan. Don't make any more. Don't make any more. So let me see if I can illustrate this. What's going to happen is the tryptophan itself is going to bind. So now when it's present, it's going to bind to the repressor protein. The repressor protein is going to block right here and therefore shut down the whole pathway. And so let's take a look at that. And so here's the regulator gene. Here's the inactive protein. Here's tryptophan. When it binds to the repressor protein, it makes it active. It attaches, when I say it, the repressor protein attaches to the promoter region, therefore blocking transcription. So bacteria are able to turn off the production of protein when they don't need it. They can repress. It's called a repressible operon because they can repress making a protein when they don't need it. It's incredible. Now, let's think about this. If they can turn off, ponder this, if they can turn off genes, maybe sometimes they would want to turn on a gene that's normally off. How might that work? And so let's look at this. It all comes down to the DNA and whether or not the repressor protein attaches or does not attach. If a bacteria produces an active repressor protein and it's attached to DNA, and then that means the genes are turned off. So then you would want to turn them on. So you need to have something attached to the repressor protein that derails it, if you will. So by binding to the repressor, an operator sequence is reversible, meaning it's either blocked or not blocked. And so at low levels of tryptophan, the repressors are inactive and therefore the operon is turned on. And so when you increase tryptophan, it turns off the operon. So we call that a repressible operon. But some operons, as they determined, are inducible. And if you're going to induce someone to do something, that means you're going to sort of entice them. In other words, they're stimulated to be turned on. And so inducible operons produce an active repressor protein. So the genes are off, and then when they need to be turned on, the repressor protein becomes inactive. Let's look at this. One of the most famous experiments they conducted is, again, they were growing a lot of bacteria. And here's a petri dish, here's the bacteria. They started experimenting with milk sugar called lactose. And so let's talk about this for a second. Lactose is something that's sort of infrequent. Milk sugar doesn't come bacteria's way very, very often. So why would you make the enzyme lactase if you don't have it, if you don't have lactose? So when you give, in other words, if you grew bacteria on a petri dish and you added the powder sugar, you added powder sugar to this lactose, guess what? They start making lactase. It's incredible. The substrate itself, a sugar, induces the production of a polypeptide enzyme, lactase, necessary for the hydrolysis of itself. It's pretty good. Pretty good. So where is E. coli? Yeah, it's in the colon. That's where it lives, and it's in here. And so it's like kind of an unpredictable environment. It basically eats whatever you eat. And so if you're going to drink milk, it's going to get milk. And so milk's going to come down. It's like, hey, she drank milk. And so therefore, now it's time to turn on the lactase. And so here's the lactose, which is the substrate, the sugar. The enzyme that speeds it up is called, let me do this, it's really big. The enzyme that speeds it up is called lactase. Sometimes lactase is called beta, because that's a beta. Beta 1,4-galactosidase. Beta galactosidase is the true name of the enzyme. Whatever, it's still called lactase. So I'm telling you, if you give bacteria lactose, they'll make lactase. If you don't give them lactose, they won't make it. So they're able to regulate this. It's pretty cool. So lactase is the enzyme necessary for breaking down lactose into 2 monosaccharides, as you can see here in this picture. So how does it work? The operon for lactose is called the lac operon, and it involves a regulatory gene, or sometimes it's called lac-I instead of lac-R. Sorry, but that's the way it is. So the lac gene regulator gene produces a repressor protein. And it turns out the repressor protein's active. And I know that because you can see it bonding right here to the operator, which means the genes are off. These are the genes. They're off. They're off. But then, someone drinks milk. Someone's drinking milk. So the lactose comes in. It attaches to the repressor protein, making it inactive. Therefore, RNA polymerase is able to bond to the promoter, move past the operator, transcribe the genes, and produce the enzyme lactase and a little, not enzyme, but a protein called permease that helps more lactose get into the cell membrane. I'll show you a video of that in a moment. So it's most incredible. The regulatory gene makes an active protein, which means the operon is off most of the time, but the sugar can actually turn it on. It's most incredible. So the sugar induces the production of lactase. It's remarkable. And so let's take a look at this little video clip, which I find to be very helpful. Let's see here. Here's sort of a cast of characters here. So this is what we're talking about. In bacteria, the chromosome is in one large loop right here. And so this region in the DNA is the lac operon. In other words, the promoter, the operator, the structural genes, and the terminator. They all work together. And so there's the bacterial DNA. Here's the repressor protein. It's our next member of the discussion. Here's messenger RNA, which is transcribed or not. Here's the disaccharide lactose. You can notice here. And then here are the two proteins that are produced. There's lactase or beta-galactosidase, and then there's this permease, which is really cool because that lets even more lactose in. So as it turns out, in your intestines, when you drink milk, a little lactose gets into the bacteria, just a little bit. And when the lactose gets into the bacteria, it comes along, we're talking about a sugar now, and it finds this repressor protein, which normally the genes are off because RNA polymerase can't get past this thing. So RNA polymerase is not doing anything at this point. But then when a little lactose comes in, the lactose attaches, which changes the shape of the repressor protein, derailing it, and now RNA polymerase is going to have a free time to cruise along, produce messenger RNA, there it is, and what it's going to do is it's going to go to a ribosome, so that was a little transcription. Once messenger RNA is produced, the ribosome is going to make the protein, so that's why gene control is better than translation control. So there's lactase, there's a little bit more lactase, and then here's this protein called permease. And let's follow permease for a second. The more permease, remember like a protein door in the membrane, like facilitated diffusion? If there's only a couple doors, that's going to limit the amount of milk. But say you're like drinking chocolate milk and there's a milk. The bacteria want to take in more and more lactose. So the more doors in the membrane, the more permease, the more they can take in. Isn't that incredible? They could regulate the amount of protein transports when there's more milk present. Remarkable. So now that all that lactose is present, it derails, but then once all of the lactose is digested, the repressor protein attaches back and shuts off the whole situation. And so lactose induces the production of lactase. So there's our little story of gene regulation. I hope you enjoyed it. Thanks for watching.